Well, good afternoon, everyone, and thank you all for joining us for our inaugural presentation of Urology Speaks. My name is Alan Parton, and I serve as the director of the Brady Urological Institute and a urologist in chief for Johns Hopkins Medicine. In just a few minutes, I will be introducing our speaker for today's session, Dr. Sarah Ammond, who will be leading our talk entitled Ecology Meets Cancer Biology, Defining the Keystone Cancer Cell Species to Develop a Cure. I cannot think of a more engaging topic to kick off this web series than the subject of curing metastatic cancers. This work encompasses the entirety of the Brady's tripartite mission of excellence in research, education, patient care, through the pursuit of our groundbreaking discoveries. Now, metastatic cancer is no stranger to the global public health arena. In fact, it has been a top concern for medical care teams, basic science research, and community leaders around the world for decades. This topic is truly frightening, humbling, but also profoundly motivating. I'm often reminded how the findings and treatments we develop here at the Brady Laboratories make a positive impact on even the most far-reaching communities across the world. What Dr. Ammond will soon share with you is an incredible example of how the curiosity and tenacity of our investigators, doctors, nurses, basic scientists, and staff take seed ideas and develop into groundbreaking discoveries that have the potential to change the way we understand and treat urologic disease. Normally, we would hope to be able to share these exciting highlights with you in person. After all, as a patient, donor, friend, and advocate, you are an integral member of our team. But we are adapting to the times and delighted that we live in a world with the technology to bring our amazing people and stories of discovery to your home or wherever you may be at this moment. I'd like to make a comment about philanthropy, as many of you on this webinar have provided support for our mission. Philanthropy has long been a critical part of Brady's and Johns Hopkins tradition and history. In fact, philanthropy is what enabled the establishment of our institute. Thanks to Mr. Diamond Jim Brady, a successful railroad company investor, Mr. Brady was a patient of Dr. Hugh Hampton Young's, the forefather of modern urology and a trailblazing physician from the Johns Hopkins Hospital. In 1914, Mr. Brady became a partner in discovery by making a generous gift to build and launch the Brady Urological Institute so that Dr. Young's expertise could have a more widespread impact on human health for generations to come. Now, 106 years later, we continue to pursue our mission in honor of not only our clinical predecessors, but also Mr. Brady. Similar to Mr. Brady, your support and encouragement is what fuels our work, and we did not want to let a pandemic keep us from recognizing the impact that you have on our institute and ultimately the future of medicine. So please accept my thanks on behalf of our speakers, faculty, residents, staff, and patients now and into the future. Today, we're honored to have this opportunity to connect with all of you so that you can hear directly from our experts and ask them and me your questions. Now, a few quick notes before we get started with Dr. Ammond's presentation. This afternoon's webinar is being recorded, so we will be able to share it with friends who are unable to join us. If you are interested in receiving a link to the recording, please let us know, as we invite you to pass it along to any friends in your family who might have an interest in watching it. After Dr. Ammond finishes her presentation, she and I will take your questions. Please use the Q&A button at the bottom of your Zoom screen to ask a question during the program, and we will try our best to answer as many as we can. And now, I proudly introduce you to our speaker for this afternoon. Dr. Sarah Ammond is an assistant professor of urology and oncology who joined the Brady faculty in 2018. She works closely with Dr. Ken Pienta, medical oncologist and director of research here at the Brady, to study the ecology of cancer. She completed a postdoctoral fellowship in Dr. Pienta's lab, received her undergraduate degree from North Carolina State University in biological sciences, and received her PhD from Washington University in St. Louis. Dr. Ammon has been an invaluable asset to our institute, bringing a novel and game-changing perspective to cancer research. Dr. Ammon, I'm delighted to turn this session over to you.
Thank you, Dr. Parton. I'm really excited to be with you all today. I come from the cancer question in a little bit of a different perspective. As Dr. Parton mentioned, my background actually started in broad biological sciences, and my first research experiences were actually in plant pathology. When I went to graduate school, I started focusing on the role of the host microenvironment, which has become more and more a part of how we think about cancer. When I came to the Brady as a postdoctoral fellow, I sought to bridge these two ideas. So bringing the ideas of evolutionary ecology and the background in microenvironment and how interacting cells in their environment can drive cancer. I'm going to walk you through this journey that we have created over the last several years. It's really an exciting one. We believe we've found the Keystone Cancer Cell species, that really critical cell type that may open the door to a true cancer cure. Now, I'm a basic scientist, and so I think it is especially important because I don't see patients face to face, but instead am a step back from them here in the lab to consider our ongoing health crisis of cancer. Worldwide, cancer kills 10 million people per year, and in the United States alone, that represents 600,000 people per year. Now, those numbers are pretty big, at least for me to think about in real terms. What that means is that one person is dying every minute of cancer in the US. That's 60 people lost in the hour that we'll spend together today. Now, when cancer is truly localized, when, for example, we study prostate cancer, when it's truly localized to the prostate, if that tumor can be completely removed, like during radical prostatectomy, survival rates are very high, near 100%. So what accounts for those cancer-related deaths? There are two central challenges to cancer survivorship. The first is metastasis. So when cancer leaves that primary site and it spreads to all parts of the body, in prostate cancer, most commonly the bone, that's what's known as metastasis. And the problem with metastasis is that we cannot cure cancer with systemic therapy. All cancer becomes resistant to any standard of care therapy, and that includes hormone therapy, chemotherapy. And what's really fascinating about this question from a basic science perspective is that cancer is resistant to all forms of known systemic treatment even before it's been exposed to it. Classically, these two challenges to survivorship, metastasis and resistance, are considered separate silos. They're both attributed to tumor cell heterogeneity. So within the tumor, there are diverse types of cancer cells with different genetic variation. And some of those variants may, by chance, be resistant, have a resistance mechanism to a particular drug, while other cells, by chance, may escape the primary tumor and establish in a secondary site. What's really interesting to me, though, is that these are kept separate. They're researched separately. Very little actually crosses the divide to consider both of these challenges to survivorship. Now, from an ecological perspective, an evolutionary perspective, that's really non-intuitive. Both metastasis and therapy resistance requires a high degree of resiliency. Those cells must survive chemotherapeutic assault. Therefore, we would then hypothesize that they would have the same, what we call co-adaptations, that they would have the same traits needed to survive. We're going to start with the metastasis space because from an ecology point of view, it's studied a lot. So why move? This is true of human beings, this is true of bacteria, in addition, it's true of cancer cells. Just because you can move doesn't mean you do move. Movement is incredibly high risk and it's high energy. You have to use up energy source that you could use if you were a cancer cell for proliferation. and you're exposing your cells to new risk. One of the most common stimulants to move is scarcity of resources or an abundance of toxins. Now, if we were in person at this point, I would invite you to think about a coffee break. If you had a late night last night and really need a lot of coffee, you would be more willing to take increased risk to get to that coffee cart. So even if somebody you really didn't want to talk to was standing there getting a cup for themselves, you would still go and get some. On the other hand, maybe you didn't need that much coffee, but they have really great pastry. So something else that would pull you into that site. That, what we just described, that give and pull between what you want, your positive goods and resources, and the cost associated with it, as well as your ability to get up and go to the coffee cart, is all described under optimal foraging theory. Now, OFT, optimal foraging theory, is well described in the field of ecology, and it predicts how an organism will behave when searching for resources and responding to hazards. Essentially, it balances ability, cost, and benefits to maximize fitness, to maximize survival. So why do organisms move, and why, as we found, do cancer cells move? To move toward good and away from bad. If we think about factors in the environment, in this case, the tumor microenvironment, there are many goods. There's oxygen, there's glucose, there's sugars and carbohydrates and fats to survive and proliferate and drive the metabolism of the cell. There's also bads, though. For example, chemotherapy, a toxin in the microenvironment. All of this ability to move, though, is hinged on the individual's capacity to actually move. Now, this is a problem for cancer cells because cancer cells come from an epithelial cell that is programmed to stay in place. It's how our bodies work. It's how our tissues are separated. So how does it overcome this? We can once again turn to ecology, where there's a really beautiful example of what's known as a transmogrification from a proliferative, from a reproductive morph to a dispersal morph. So you can see here a picture of a grasshopper. They're very small, very short, just a couple of inches long. And these are known in ecology as a stay-at-home morph. Really beautiful language. And grasshoppers stay at home, stay in a local habitat when there's high resources, when there is plenty of food for everyone. If, however, there's overcrowding and reduced resources, as a grasshopper develops, it will transmogrify instead into a dispersal locust. And of course, locust plagues were in the news over the last few months. But what's really important about this is that these locusts, they can't reproduce as quickly, but they can travel hundreds of thousands of miles. They're much larger than a grasshopper. They need longer wings to fly. What's really fascinating about this story of our grasshopper and locust is that these two pictures are actually siblings. They're twins. They have identical genetics. It's only a change in their circumstances, a change in their gene expression and their phenotype, so how their body plan is organized and what they are able to do well, is all due to their environment. This led us to the question, well, what about cancer? Does it have a grasshopper and locust morph? To answer this question, we turned to a prostate cancer cell line model called PC3, and we used a microfluidics device. So it's very small, it's about the size of a dime, and these cells are labeled green and red. And this microfluidics device allows us to place a gradient. So in this case, we are testing the ability of cancer cells to respond to high-dose docetaxel, which is a common chemotherapy in prostate cancer, to low doses of docetaxel. And then we track these cells, and I'll be showing you a movie here in a moment, over several weeks. So we're going to let this play, and the first thing I want you to notice is that we have die-off of the cells under the high-dose docetaxel. It's really good. That means our drug is working. This is why docetaxel works in patients. So as you can see first that the drug works and the cells die, what becomes immediately apparent are these very large cells, here in green, the red ones do it too, that are moving around rapidly, even in the high-dose docetaxel. And then eventually, as we see here, they seed what we can consider an in vitro recurrence. So small cells then repopulate that space. This was frightening to us because we were watching the acquisition of therapy resistance in real time. But it was exciting because it also meant that we made a critical discovery. And what I'll be telling you a little bit later, perhaps more of a rediscovery of what we call the polyaneuploid cancer cell. So this is a polyaneuploid cancer cell you can see on the right, this very large green cell. Typical cells, it's parental cells, you can see in these red images here. So you can see there's a big difference in size. In addition, as I'll be showing you, they have an increase in DNA content. What we could observe from this movie is that these cells are highly motile, they're highly resistant, and they seed recurrence. Interestingly, these multinucleated and polyploid cells, so again, that means increased DNA content, have been reported in the literature for decades. They've been described after therapeutic intervention in multiple cancer cell types. And those interventions include different types of chemotherapy, radiotherapy, and even hypoxia, so low oxygen in the tumor microenvironment. What's interesting, though, is that it was long assumed that these cells couldn't survive. They were too disorganized. They couldn't proliferate to form two smaller cells, and so therefore must die due to what's known as mitotic catastrophe. So we had to prove to the field and to ourselves that this wasn't just simply an artifact of in vitro cell culture. We asked the question, do these PACs exist in human tumors? It's often said that a day in the library will save weeks in the lab, so we hit the books. And as it turns out, the first description of one of these cells was actually in 1858 by the father of cellular pathology, Rudolf Virchow. And he looked at tumor slices. So he took tumors after autopsy and sliced them and then looked at them under some of the very early microscopes. And right away, he observed these various polymorphous cancer cells. And you can see his beautiful illustrations here on the right that clearly show very similar types of cells that we saw in our microfluidics device. Well, what about in our animal models? Can we really start to study this? What this is a picture of, it's known as an H&E stain. The dark blue is nucleus, or DNA, and the pink is cytoplasm, it's the rest of the cell body. And these are human prostate cancer cell lines that are grown in the back of an immunocompromised mouse. And what you can see in these circled regions are that we were very quickly, once we knew to look, able to pick out PACs even in these tumors. Okay, what about in human tumors? This is an example of a metastatic lesion. And this time, the blue again is DNA, but we included a cell border stain to make sure we were actually looking at DNA all within one cell. And what you can see very clearly in the slide, indicated by the arrows, are a number of polyaneuploid cancer cells. So now we know that Virchow found them in a primary tumor. We found them in a metastasis. Well, are they getting there? Are they the cells that are leaving the primary tumor and going to the metastatic site? The answer is maybe. This time we took peripheral blood from a patient with metastatic prostate cancer, smeared it on a slide and then stained in order to see various markers. So what we can see here in the middle is a cancer cell, and then these are normal white blood cells. But what I hope you can appreciate right away is that here is a cancer cell with 3 distinct nuclei. We have another one on this side that has multiple nuclei as well. Now this was a bit humbling, to be honest, because most computer algorithms, for all this time that we've been studying rare cells, are trained to exclude cells that have funny looking nuclei. So we've been missing these critically important cells the whole time. We next wanted to know whether this was a prostate cancer phenomenon or whether it was universal to all cancer cell types. And I'm not going to go through this data very carefully, but this is a summary table showing you that we have found PACs in both cell lines and pathology from breast, colon, ovarian, lung, glioblastoma, and bladder cell lines. And right now, in our incubators across the hall, we have head and neck cell lines growing, and we're planning to check those in the coming weeks. So now we know that PACs are relevant to human cancer and not just cells grown in a lab in vitro. So what have we learned about PACs? What you're looking at here is a rotating view of a three-dimensional image that we now can take due to confocal microscopy of a normal PC3 parental cell and a PAC on the right. And what I hope you can appreciate is, again, the blue, in this case, again, is DNA, is that in the normal cell, it's nicely organized and packed into this nice tight nucleus with clear margins, versus the PACs on the right that have clearly disordered nuclei. They're morphologically very different. In addition, these two images, as they rotate, are on the same scales. So we right away appreciated that these PACs are much larger, not just in 2D, where we sort of saw them flat on a piece of plastic, but also in 3 dimensions. Now, we did put some numbers on this, which is important, not just to take a qualitative view. So we looked at cell volume, and again, this is the parental cell versus the PAC, and we found that on average, PACs are about 5 to 7 times larger in volume than their parents. And we also looked at DNA content, known as ploidy, so how many copies of their complete genomes do these cells have? The parental cells have a peak at what we know as 2N, so their regular amount of DNA, and then right before cells divide, they double their DNA in order to make 2 cells. There's a smaller peak there. What's incredibly striking about the PACs is that there's an accumulation in this at least 4N peak. So they're pushed over to a higher degree of DNA content. We also found that more PACs are formed after treatment, and that this doesn't matter what type of treatment or type of cancer cell line. These images on the left are what is known as phase contrast, and this is our control. And I'm going to outline these cells here on the left, and you can see that they're quite small. In contrast, after only 72 hours with chemotherapy, all the cells that survived were these giant PACs that I'm highlighting on the right. And really interestingly, whether we treated cells with docetaxel, etoposide, or cisplatin, that all work in different ways, and no matter what cell line we checked, we saw an increase with the increasing amount of dosage. Now, one of the first questions we get is this just the small cells all dying off? Because we know that there is at least one in this picture, big cell, remaining. Well, actually, there's a dramatic increase in the total number of PACs, 10 and sometimes 15-fold. So when we seed cells, when we seed 1 million cells, there's about 30,000 PACs there just at baseline. After only 3 days in treatment, there are then at least 30,000 PACs. So there's this dramatic enrichment of this novel cell type. How do PACs form? I talked to you a little bit about how one cell doubles its DNA and then divides into 2 equal cells, but then how do you get one big cell? One way this might happen is by undergoing late endomitosis. So what that means is that the cell starts to go through its normal cell cycle, it doubles its DNA, and then it fails to divide. So we'll walk through this video here, and we're going to be looking here. A cell starts to divide, it balls up, and you have 2 cells, and then they seem to bounce back together again. Now this cell is now a PAC, and conventional wisdom would tell us that that cell can no longer survive, but in fact we tracked the cell for weeks, and it survived. What I hope you can see from this movie right away is that, contrary to what we thought, that these cells are dying and are sort of still on the plate and not doing much, they actually have remarkable motility and morphological differences. A great graduate student here is starting to put numbers on this. Here again are movies. We have a PC3 parental cell line and then a PAC population. And I hope you can see it in the movies, but if you can't, we quantitated it here in the graph on the left, and you can see that PACs have this striking increase in motility. So how have we missed these cells for so long? Virchow described them more than a century ago. Why hasn't this been the focus of all cancer biology research? Well, for one, it has to do with our training and our bias. So again, I introduced this picture before, that this is a standard cell culture cell line. And what I was taught as a cell biologist, and indeed what I taught others until we made this discovery, was that that cell was unimportant. It would die. We don't need to worry about it anymore. Alternatively, we've become overly reliant as a field on these plate-based assays. So instead of looking at the cells through the microscope, we put chemicals on them so we can tell whether a cell is metabolizing something in a particular way or whether it's doubling its DNA in a particular way. And so this picture of cells very much alive on the plate was actually taken at what we would typically consider about a 20% viability. So we've stopped looking at our cultures as a field. Now, this brings us a little bit back to the question about whether PACs are artifact. So even if we found them in patients, even if they move more, do they actually actuate disease? Now, most scientists assume that they're evolutionary dead ends, that they're destined to die. And if it's true, it would imply that PACs may be a biomarker of stress or cancer progression, but that they are not actually contributing to the process itself. So far in this story, we've learned that PACs are resilient cells that form in response to stress, that can survive anti-cancer therapy, and that are highly motile. So that means that we definitely have a locus morph in hand. The question is, can this locus morph switch back to a grasshopper morph? Can these PACs actually seed a recurrence and form a recurrent tumor following therapy failure or at a metastatic site? To our surprise, that answer is yes. PACs can give rise to this recurrence of typical size cells. So this is PACs back to normal cancer cells, not PACs into PACs. Here we have PACs that were induced with cisplatin, and you can see that this is a whole plate full of those very large cells. We stuck these cells in the incubator and largely ignored them. So we gave them normal media, so it doesn't have any chemotherapy, plenty of nutrients. And we left them there for 75 days. And when we came back, the culture flask was full of these very small cells. Now, of course, some of these PACs remain, but by and large, recurrence isn't made up of big cells. If you remember back to those slides of tumor samples that I showed you, the majority of the cells weren't those big cells. It's a minority population. It's a very critical minority population. And here in the back, I just show you this picture to prove to you that I didn't just cherry pick this smaller image here where you can really see the cell borders. This was the whole flask was now back to normal. So we talked before about the importance of size, but what about the importance of DNA ploidy? You've already seen these first two graphs. We have DNA content across the horizontal axis, and we have parental cells that have normal amounts of DNA and cells undergoing cell division that have a little bit more DNA. When we added treatment and got PACs, this number increased. So most of the cells had more DNA than usual. After a recovery period, however, of those 75 days, the DNA content distribution goes back to normal, where most of the cells have a normal amount of DNA again. One of the reasons why this is interesting is that it may be a point of therapeutic intervention. So if we can keep these PACs just in their PAC form, and they never start to divide again, then that means that we can stop recurrence. Now, PACs may repopulate through a number of different mechanisms. One is by meiosis or budding. So this is a still image of a PAC, and another cell may be blebbing off here. The other is by asymmetric division. We have a PAC in the middle of the screen here, and in a moment, instead of dividing into two, it's actually going to divide into 3 viable, happy cells that go on to live for weeks. So what we know about PACs: they're found in human cancer. They're increased in size and DNA content, and that may be important for their function. More are formed in response to treatment. They have high motility, and they can seed a recurrence. So all of these components are really coming together and led us to our hypothesis that PACs are the keystone species of the lethal tumor ecosystem. I think one of the most common keystone species are the wolves of Yosemite, of Yellowstone. And this is a beautiful story where they took away the wolves and the ecosystem started to collapse. Different populations were more and greater, and even the vegetation changed. They reintroduced the wolves, and within just a couple of years, that ecosystem found its equilibrium again. So a keystone species is one that plays a critical role in maintaining the ecosystem with a very large effect despite a very few number of cells. And it actually gets its name from the architectural keystone itself, and a keystone supports two arches, two walls of an arch, or the dome structures of some of the amazing cathedrals. This led us to a paradigm shift. And for a cancer biologist, this is a really big deal. Instead of considering metastasis and therapy resistance as distinct siloed fields, we instead began to think of them as both supported by the polyaneuploid cancer cell, by this PAC, what we now identify as the Keystone Cancer Cell. Elimination of the Keystone Cancer Cell may then be the answer to a cancer cure. If the PAC is the keystone species and we eliminate the PAC, then the lethal functions of metastasis and therapy resistance are no longer supported, which would lead to collapse of the cancer cells and the tumor itself. So how can we do that? I already talked to you about how standard chemotherapy forms PACs, but the important part about this is that it does reduce total tumor burden. That's why it works so well in patients. So it reduces the total tumor burden, but enriches for PACs, which provides us a window of action. What we propose is an evolutionary double bind treatment strategy. So double bind is sort of out of the frying pan and into the fire. So if we treat with this cytotoxic therapy to kill the majority of the cancer cells and induce these keystone cancer cells, then we come and treat with a therapy that will specifically eradicate these cells directly. This begs the question, what is unique and potentially targetable about these keystone cancer cells? To begin to answer this question, we took a global approach. So what you're looking at here is what's called a heat map. It's called a heat map because the orange or the warmer colors mean high and the cooler colors mean low. And this is for RNA, and in our cells, RNA, it comes from DNA, RNA then encodes all of the proteins needed for life. So we have these parental cells, and those are the same parental cells that we've been talking about this entire time. And then the three right columns are all PACs that are induced by 3 different therapies. And now it doesn't really matter the details of this. But what's important is that if you step back and take a look at this, that the control population is quite different from the PACs. That means that they have unique vulnerabilities. Highlighted in yellow are areas where we're already starting work now to try and identify what specific pathways or specific proteins to go up against. A few vignettes though. One is that we found that keystone cancer cells may survive by going into a hibernation state, similar to bears in the wintertime. So this state of dormancy or quiescence is when the cell stalls in cell cycles, sort of steps out of cell cycle and protects its DNA. To get this image, we engineered cells with a special cell cycle reporter called a Fuji cell reporter that turns red when cells enter this quiescent state. We're working on this now with even newer reporters and even more sophisticated methods to see if all PACs go into hibernation and to start to study how they enter and how they exit. We also now know that keystone cancer cells have increased fat stores. What you're looking at here in green are lipid droplets that are accumulating in these PACs. We believe that keystone cancer cells may use these fat stores to survive while such stress is present. I, of course, don't have time to go over all of this today, but this is our overview of how we're thinking of attacking PACs. You can see that we're thinking of a number of different mechanisms, including their metabolism, their ability to form recurrence, and how they're responding to stress. A really exciting part of this evolutionary double-blind strategy is it means that we can repurpose drugs that have already been approved. And these drugs in yellow are those that are already approved for other indications. In summary, we have much to learn about these keystone cancer cells, and it really takes an ecosystem to do this work. This high-level discovery and inquiry requires powerful tools. I've shown you data from our Sprint nanostring machine and images from our confocal microscope that allows us to look at PACs over time and in 3 dimensions. Coming soon, very soon, in the coming weeks, is a machine called an IncuSite that will allow for very long-term cultures over weeks and months with high-power imaging for us to get an idea of what this kinetics looks like. And we are grateful for federal grants and foundation and private funding to make all of these tools possible. The other thing that is essential about this is truly collaborative and interdisciplinary scientists. We work with scientists from around the world, and now with Zoom, we can travel around the world in a single day, drawing from expertise so that we can learn from the best. You are a part of our team as well, and thank you for letting me share this story with you today. All that is left is for me to thank our SuperPAC. On the left here are current members of our team, including Ken and our many collaborators, both in Hopkins and beyond, and special thanks to all of our funding sources that made this work possible. And with that, I am happy with Dr. Parton to take any questions that you have. Thank you.
Thank you so much, Dr. Ammond. As a reminder, please use the Q&A button at the bottom of your Zoom screen to ask a question, and I will read them off as they come in. The first question that we have received, Dr. Ammond, what has surprised you the most when researching these cells?
I think a big surprise to us was just how resilient they were. As I alluded to, cell biologists have thought for decades that these were just dying cells. So that cells survived such stress and then gave rise to other cells was absolutely remarkable. And it took a while and a lot of data and careful science in the lab to really convince ourselves of that truth.
Thank you very much. There's a question here. Are PACCs and KCCs the same thing? And if not, how do they differ?
Yes. So we believe that KCCs, Keystone Cancer Cells, and PACs are the same thing. That PACs, as polyaneuploid cancer cells, those really big cells, are the keystone species of the tumor.
Dr. Ammond, how does immunotherapy come into play here, if at all?
So we're not quite sure yet. We are working to understand very specifically how these PACs will respond to immunotherapy. I think it's a really great question, and it's something that requires very careful interrogation. So we hope, and all signs point to, but we still need to test this, that they may be, T cells might be able to kill these cells more readily because they look so much different from normal cells, and that immunotherapy would help in that space.
Dr. Ammond, you spoke in terms of cancer cells seeking out an ideal environment or increasing movement in response to scarcity. Can this information apply to lifestyle as a way to decrease the risk of recurrence or metastasis? For example, would a low sugar diet discourage the movement of cancer cells? Or alternatively, would intermittent fasting encourage cell movement?
So it's important to recognize that what a cancer cell sees is only its very local environment. And by the time the circulation is getting to the cancer cell itself, it's not necessarily going to recognize your high sugar, low sugar, or fasting state. So our work does not address any of those lifestyle changes at all because we are only at the cellular level.
Do we know if PACCs express PSMA?
We think they do. That's actually a question that is actively ongoing in our lab right now. We have a really great person who is trying to understand whether PACs, like other cancer cells, express PSMA. We know that it's not only PSMA expressing cells, though, because this comes from, we see PACs in PSMA non-expressing cells as well. But a really great question.
Dr. Ammond, as Keystone Cancer cells seem to apply across the cancer spectrum, what progress have you and your team made in bringing other researchers from other cancer disciplines aboard to increase the momentum?
So we're actually doing a lot in that space. So we are trying to tackle the urological cancers here, of course. We have collaborations with breast cancer biologists, in particular, that cell cycle, beautiful image with the Fuji cell line. That is work done in a breast cancer space. I'm meeting with a past collaborator who studies multiple myeloma next month. We have also moved some into the lung space with researchers here at Hopkins. So we are moving with great momentum.
Do PACCs still need TME to survive, or have they become truly autonomous?
Right. So TME there, I think, refers to the tumor microenvironment. And we know that PACs are formed in the tumor microenvironment, but we believe that those PACs then can go to another environment and be successful there. So we believe that it's needed for that initial formation, but not for eventual survival.
There's an interesting question about related to active surveillance, and maybe Dr. Parton, you'd like to answer this. Do we know if this research has implications across the prostate cancer spectrum, and particularly for patients who are undergoing active surveillance? Might it help to identify patients who are good or poor candidates for treatment intervention?
I mean, that's a great question, actually, Alyssa. The majority of our patients that we put on active surveillance are very far to the left of the spectrum when it comes to developing metastasis and resistance to chemotherapy. But I think that if these cells are able to be identified, and actually you could see them on a prostate biopsy at the time of diagnosis, there is a future for this being a prognostic marker as well.
Are nanoparticles a possibility to use to be a systemic treatment for any of this work?
So nanoparticles are great. And for those who don't know what a nanoparticle is, it's a small sort of package to put in a higher payload of drug that can theoretically then be specifically delivered to a particular site. So in that respect, yes, if we knew how to deliver it. I am working closely with Stavroula Sofu, who studies nanoparticles, and she's an expert in membrane dynamics and how to deliver those specifically to the cells that we want to kill. So yes, but not ready for prime time yet.
As you pursue all of your ideas to attack these keystone cancer cells, are there any clinical trials using some of the drugs you suggested going on right now? And if not, how soon, what does the timing look like for clinical intervention?
Right, so many of the drugs that we're interested in using have gone through clinical trials and are known to be safe in patients for other indications. So for example, for diabetes or high blood pressure. So we know that the drugs are safe. These drugs have not been specifically tested in cancer necessarily, and it's going to be important with our evolutionary double-blind strategy to consider the timing. So that first you need to be treated with chemotherapy, followed by the second intervention. We are hoping to get those clinical trials up and running in the next 2 years.
I think, Alyssa, at one point, I sit on the porch and drink coffee with Ken Pienta a couple times a week. One of the points that a lot of people may miss is that it may not just be important to kill these cells, but to actually keep them from dividing. And the timing, I think, that Sarah keeps mentioning is what the critical aspect is going to be. And I believe Sarah can tell us they may have identified a couple molecules that might be specific for these cells.
Yeah, so we have a couple on our short list. So in particular, thinking about how those cells divide, we're interested in understanding, it's a protein called KIPC1, for example, that's really important for that unequal cell division that we saw a movie of. But again, that timing is going to be critical.
Someone has submitted a question asking if we know if there's a connection between these Keystone Cancer cells and BPH.
I think that's a great question. So far, we haven't found any evidence of these Keystone Cancer cells in the BPH setting. I would hypothesize not. So we know that normal prostate doesn't typically have this type of morphology even at a single cell level. It's something that we need to go back and test very specifically, and we're working with Angelo DiMarzo here, who has done a lot of work in that space. He's an excellent pathologist to look at prostate cancer sort of along different stages of progression, along different Gleason grades, as well as those states like BPH and normal prostate as well. So we are looking, but we don't think so.
Dr. Ammon, do we know if hormone therapy might induce the formation of Keystone Cancer cells?
Short answer to that is yes. Hormone therapy will induce formation of PACs.
In any of your current research on the Keystone Cancer cells, have you been able to eradicate them?
So we can in a dish, which is a lot different than doing it in an animal or in a patient. So the levels of drugs that we have been successful in so far, killing the PACs directly, are much higher than what a patient can see. What we have been able to do is, using this evolutionary double bind strategy, is keep them in that PAC state and not allow them to divide. So again, in experiments ongoing, we have stopped them and sort of frozen them in that PAC state, and now we're waiting to see if they ever start dividing again. But we need to wait a long time to be sure of that.
Since variation drives evolution, there's a question about perhaps there are other metastatic drivers in addition to Keystone Cancer cells. Have you eliminated that possibility?
Yeah, so I think the question is alluding to the sort of genetic heterogeneity and the known genetic drivers of cancer. And it is undoubtable that there are many routes to metastasis through a genetic and a very specific variation in particular genes. But what has been really compelling about PACs is that we have found it in every cancer type. We have found it in response to every therapy. For any of these drivers that have been identified before, they have been specific, either to a particular drug. So if a cell upregulates a particular drug pump, for example, due to its genetic variation, it then is not also resistant to another drug. These PACs we found are true across drugs, and the same holds true across all cancers. So it's not that those other factors aren't at play, but we believe that the PACs are sort of a unifying factor in all of these different cancer types and across all of these different therapies.
Do Keystone Cancer cells have an oncogene or oncogenes, or in other words, genetic heterogeneity?
So what's interesting about these PACs, again, is that they arise from all these different types of cancer. So we have formed them, for example, from a breast cancer cell line that has its own unique set of genetic variants, and a prostate cancer cell line that has a different set of unique variants. When we form PACs, they don't seem to change that genetic material in such a dramatic way. Unlike, for example, person-to-person variation of breast cancer, where one person may have one gene that really started the cancer and another person may have another gene that really started that cancer. Here, the PACs, it really seems to be a factor of forming that cell type versus what in particular is going on with the DNA. Now, what goes on afterwards, we're still trying to figure out. So whether the cells that come after the PACs are somehow genetically different than the ones that came before.
Dr. Ammond, is there a thought that if we believe cancer is eradicated in someone and perhaps there were dead cancer cells, that in reality, the cells could come back to life?
So it's not that these cells can come back to life. We know that cells can lie dormant, so they can be asleep for a very long time, sometimes decades, without then waking back up and starting proliferation and actually forming a tumor that would have an impact on a patient's life. It's not that those cells are dead and then came back to life. It's that they were asleep and woke up.
From a clinical aspect, we, thanks to Dr. Walsh's careful monitoring of his patients, we've seen men that had undetectable PSA values for 16, 17, 18 years, and then suddenly they begin to have a measurable level. So that tends to make me believe that what we're seeing here has a clinical application as well, dormancy.
We received a great question for both of you. Has there been any evidence to show that surgical treatment of prostate cancer may induce the development of keystone cancer cells? And if not, is it something worth studying?
I can go ahead first. To my knowledge, there's no evidence that surgery induces the formation of keystone cells. There is some vague and controversial evidence and suggestion that surgery could release cells from the prostate while you're manipulating the gland. If that were the case, I would expect we would see many, many more men recurring than we do, but it is something that's being investigated. I did my PhD for years studying in a very crude way compared to what Sarah's doing, whether or not the surgery released cells into the men's bloodstream. And thank goodness we didn't see it very often.
Does all cancer treatment increase keystone cancer cell activity? Might there be any treatments that show less activity or less impact on keystone cancer cells? Could less treatment perhaps be better for longer survival?
So I'm going to take each piece of that question at a time. Every therapy that we've tested induces PACs. So drawing off of that, I would then sort of speculate that any other chemotherapies or poisons to the cell would also induce PACs. It's also important to note that we can make them other ways. So if we put them in 0% oxygen, for example, they're formed, or if we put them in highly alkaline or acidic conditions, change their pH, they're also formed. So it seems to be a stress response. The second piece of that question was about should patients be receiving less chemotherapy to sort of extend the time to create PACs. And here, remember that even a little bit of chemotherapy can induce PACs, and chemotherapy works really well. It gets rid of most of the cells, and it really shrinks down tumor size, and that's really powerful. There is some studies in the evolutionary dynamics of cancer, it's called adaptive therapy, about whether the time, again, it's timing though, not dosage, so whether the timing of therapy should be changed to control tumor burden for a longer period of time.
I think we have time for one more question. And you touched on this during your presentation, Dr. Ammond. Why were keystone cancer cells, or PACCs, so ignored up until now? And because you really are pioneering a new field, are you meeting resistance and skepticism from within the cancer and research community?
We meet a lot of resistance and a great deal of skepticism, which is really great in the scientific world to be met with that resistance, because it means that we need to prove our point better. And that's one reason why we have so many careful studies to assess these PACs and determine whether they are really actuators of disease or just something we see in cell culture. The reason why we have ignored them for so long, number one, they're rare. So if you don't see them very frequently, I think it's human bias to think that then it's not very important. The second reason is because they arise, again, going back to what I was talking about the changing pH, they arise under stress. So if you consider cells in culture, we work really hard to keep those cells, we call them happy. We want all the conditions to be right so that they will keep growing on plastic, which is sort of amazing in and of itself that we can grow cells in the lab at all. And so when you see something that is aberrant, that's different from what you would usually see, you think it's something wrong, you think it's an error. And because it didn't happen that often, unless you really go and look at for that question in particular, people just dismissed it.
Thank you so much. That's all for the questions.
Thanks, Alyssa. I want to go ahead and formally close us out for today. First of all, what an amazing presentation. And thank you, Sarah, for joining us this afternoon, for presenting your research team's discovery so beautifully, and for answering the many excellent questions we had, thanks to the audience. I'd also like to give a special thanks for all of you who participated in this webinar, and for your continued support of the Brady and Johns Hopkins Medicine and the dynamic collaborations between our community of investigators, clinical staff, and patients that gives me so much hope for a brighter and healthier future. If you enjoyed today's session, please consider joining any of the other sessions in the series. You'll see them on your original asks. Our next one's scheduled for Thursday, November 19th, when two of our star urology residents will present on the prevalence of prostate cancer in the Black community and our efforts to further understand the causes of the disparity and develop strategies to better serve these men. Upon completion of this webinar, you're going to be directed to complete a short survey. Please let us know what you thought of today's presentation so that we can refine the future's planned presentations. I also want to thank Alyssa Cole and her development staff for making this possible today. And overall, thank you again for spending your time with us, and stay healthy and well.